What are you doing?
I'm from Velu, sir.
Okay.
My name is K. Lakshmi,
Okay.
DKM College for Vellore,
Okay.
MSc Microbiology. Yes,
Which
sir.
MSc? Which year?
First year, sir.
Good, good, good. And then Yogashri, are you joining for the first time? Same goes for Kritika as well. Kritika, are you joining for the first time?
Yes, sir.
Okay. Could you please introduce yourself?
I'm Kritika from Vellore BKM College for Women.
Okay.
First MSc Microbiology.
Okay. Rehan, I already saw your mail. Actually, you mailed me right last time. Okay, I will show you the website directly. That's why I didn't reply. It's called Biotechnica. Okay, I will just
The
show you.
spelling?
I'm just typing it. My screen is visible now.
Yes, sir.
Yeah, here you see biotechnica.org. Okay, if you subscribe here, then they
Okay,
will
sir.
send you
Okay.
the mail regarding this. But it's a private organization at the end of the day. Okay, you might be getting lots of emails from them, so just make sure that what are you doing. The other way of looking for internships. See here, you see na internships. You can see na the Biotechnica website. There is internship. You people can see the website. What I'm trying to say, this website is to just to see the different opportunities and everything. So last time I was explaining about how to get internship and everything. So I'm talking about this website. Yeah. Shall we get into the class today? Shall we start?
Okay, sir.
Yeah, we'll start. Okay. So it will be around 1 hour class. I feel I have prepared in such a way that it will come 1 hour. Then I will be having some questions, which is already having some answers. So will help you to do some work. Okay, in the current assignment. So largely, we have seen in the last 2 weeks how the immunology people have started in a historic ways, and then we got into a broad area of innate and then adaptive immunity. Then now we are going to start the cells of the immunity. Okay, so this part whatever I'm going to take today, it's really important. The reason is there are multiple cells are there in our body. It starts with muscle cells, rbcs, then it can go up to immune cells, cd4 t cells or cd8 t cells, mast cells, natural killer cells. So n number of cells we have, including the nerve cells. In eye, whatever it is there, the retino-related cells will be there. Keratinocytes will be there, which is in the skin and then the nail and those things we will be having. But each cell will be having unique functions. Okay, so that is what we are going to see today. If you see number of cells in immune itself, it's vast. It's a huge topic, but we are going to see certain cells which are regularly used in our body, and then in terms of your exam point of view also, these are the major cells that we always study. Okay, so welcome to the week 3 lecture, and I'm Siranjeevi Rangaraj. I'm a PhD scholar in the Department of Biochemistry, University of Hyderabad, and also I'm a Prime Minister Research Fellow. To finish my teaching assistantship hours, I have been associated with NPTEL, and we are in the week 3 lecture. So, sorry, it's not one, it's week 3. So the course I have associated this year is host pathogen interaction, which is already almost called as immunology. So the code is NOC 25 BT21. So we'll start as usual. We'll do the recap on the week 3 concepts. Then some practice questions we will do. Then the doubt clearing session. If you are having any doubts, then we can do the doubt clearing session. Okay. In between my voice is not clear or something, then you can stop me at any point. Okay. So this is what Professor started. Some blood cells in the body that we have. Mostly he was talking about the erythrocytes, and erythrocytes we know that is a red blood cells, or we cannot call it as cell uniquely because it's a red blood corpuscles. The reason is this lacks the mature erythrocyte lacks a nucleus. Okay, so that's why we call it as red blood corpuscles instead of a cell in the erythrocyte case. So the we know that average lifespan of any RBC is 120 days, and which is in a bioconcave shape. The specialty of this shape is also all over the body it can go, and it cannot be ruptured that much easily. That's the reason for the shape. And other blood cells, if you see, in terms of monocyte or lymphocytes, these are the immune cells that we majorly talk, or white blood cells, basically neutrophil, eosinophil, and then the basophil. And the monocyte will become macrophage in tissues and sometimes in the bloodstream as well. Then there are other blood cells we usually study, those are platelets. So platelets are really helpful in the case of blood clotting. Okay, so if you have very low platelets, then the blood clotting will become really difficult. And in terms of dengue virus patients, the platelet counts really goes down. So the at last at the end, people will do the blood transfusion to increase the platelet count. So that will help the person to recover from the disease easily. Okay, so we are going to see majorly in this lecture. We will be studying about monocyte, lymphocyte in the sense T and B lymphocyte majorly, neutrophil, eosinophil, basophil, and macrophages, and mast cells also we are going to check. Okay. So overall, if you see, these are the generation of different blood cells from the hematopoietic stem cells. We have already studied about hematopoietic stem cell, which are pluripotent in the property wise in the previous lectures. So it has the self-renewing property as well as it can become two different progenitor cell line. One is a myeloid progenitor, the another one is a lymphoid progenitor. So the myeloid progenitor will give rise to the multiple cells. For example, it can give to the rise to the granulocyte, monocyte progenitor, basophil progenitor, megakaryocyte progenitor, or eosinophil progenitor also will be coming. So I don't know why people are joining very late. Now to change the settings of this. Yeah. Okay, so finally, this from the myeloid progenitor capacity. See, this is based upon the maturation of the cell. For example, I will ask you in a very single term. From milk, what are the products that we get? Anyone can answer. We have milk, right? So from milk, what are the different products that we get?
Curd,
Curd.
cheese,
Okay, only two. Ghee, paneer, milk gova. So there are different items that we can prepare, right? So same way, here the hematopoietic stem cell is nothing but a milk. I'm just telling it in a layman term to understand for in a general perspective. So if you see this hematopoietic stem cell will be there. So from there, it can divide into two parts. One is a myeloid progenitor, another one is a lymphoid progenitor. Okay, so then after lymphoid progenitor, it will become sorry. From the myeloid progenitor, it can give rise to dendritic cells, monocyte, neutrophil, eosinophil, basophil, or platelets or erythrocytes from the myeloid progenitor case. And also, if you see the lymphoid progenitor case, then it can give rise to the natural killer cells or T cell progenitor. It can give rise to T helper cells, and then it can give rise to TC cells, which is the cytotoxic T cells. Or else it can also give the B cell, and finally the dendritic cells also can come from lymphoid progenitor. So it's a really important 1 mark in any case. Which cell is coming from both the myeloid and then lymphoid progenitor from this picture? If you see the picture, there is one cell type which can come from both the progenitor, myeloid or lymphoid progenitor. Which cell type? See the cell type, na? Which cell type can come from both myeloid and then lymphoid progenitor? Is it audible?
Yes, sir.
Yeah. So which cell type can come from both myeloid and then the lymphoid progenitor? See the picture. I have given the picture already. My PPT is visible, right? Am I audible? My PPT is visible. Tell me.
Yes, sir.
Okay, you can see, even though if you tell wrong also, it's okay. My job is to correct you and then give the content properly. Okay, so see the picture I have given you. So my question is very simple. There is one cell type. See, for example, from milk, what you said, it can give curd, it can give cheese and everything you said, na? The same way,
Yes.
this hematopoietic stem cell is giving myeloid cell, and the hematopoietic stem cell can give the lymphoid cell. From the both the cells, which one cell type can come? Like from the myeloid side also, from the lymphoid side also. It is dendritic cell.
Dental.
See, dendritic cell here. All other cell types are can come from only one progenitor cell, whether it is lymphoid or the myeloid. But if you see overall, both this dendritic cell is the only cell which can come from both the lymphoid origin as well as the myeloid origin. Okay, so this is a really big 1 mark. And one more question in any exam they can ask, which are the things coming from myeloid progenitor, or what are the cells coming from the lymphoid progenitor? That is also one kind of questions. So the one more classification from these cells, we can say there are granulocyte cells or agranulocyte cells. So here neutrophil, eosinophil, basophil are granulocyte cells, whereas T helper cells, B cells, CTC cells, and monocyte and macrophages are agranulocyte cells. So which means granules means there are certain molecules will be there inside the granules, so which will be helpful for the killing of the pathogen or the elimination of the pathogen from the body. Okay. So overall, if you see if you want to do research on these kind of cell lines, so then you have to isolate the cell line called peripheral blood mononuclear cells, which is nothing but called PBMC. There it is. Not able to admit. One minute. Not able to admit that. Someone was trying to join, but now I'm not able to admit. It's okay. So if you see what is PBMCs, PBMCs are composed of lymphocytes, which is around 70 to 90 percentage, and then the monocytes, then the dendritic cells. Okay, so usually how people will derive this kind of blood cells or this PBMCs. So we'll take the blood from the patient or any kind of volunteer, like from you or from me, we can take the blood. So after taking the blood, suppose if I'm taking 5 ml of blood, it should be taken in anticoagulant tube. Uh, 1 minute, please. Already made. So you will take 5 ml of blood in any anticoagulant tube. Do you know what is the anticoagulant? What will happen if you don't add anticoagulant? In a normal case, you people also go for blood test and everything, na? So do you know what is anticoagulant? Yes or no? Then I can explain.
To prevent clotting, sir.
To prevent
Blood
clotting.
clotting.
Okay. So do you know any agent that useful for anticoagulant? Have you
Sir?
studied? Do you know any agent to do that property, anticoagulant property? Any agent? If you don't know, then I can tell you. Don't worry. You people are not able to recall. Okay. Anticoagulant, whatever she told is correct. So anticoagulant is to prevent the blood clotting. Okay. So there is a one big anticoagulant. We always say it as EDTA. Okay. So EDTA is EDTA, fluoride. Those things are always used for anticoagulant in the blood test. So we will take the blood. So we'll let us take 5 to 10 ml of blood if I'm taking, then I would be adding around same equal volume 5 to 10 ml of PBS, phosphate buffered saline, or the respective media. We usually grow the cells in DMEM media or RPMA. Then finally we would be adding this histopac 1077, okay, which contains a polysucrose as well as sodium triosate. So then we will do the density gradient centrifugation. So this is how it looks when you add the first blood plus the equal volume of PBS or the media, then fecal histopac, then this would be the state. Once if you centrifuge it, which is called density gradient centrifugation, then this would be separated into 3 or 4 phases. The first phase would be the clear phase, which is called plasma, and there is a puffy coat place will be there where the PBMCs will come. Then the other phase is called histopac, the whatever that we have added, and finally the red blood cells will be there in the bottom of the tube. So this is how the centrifugation process takes place. And overall, if you see, the main reason why PBMCs are coming separated on the basis of this FICOL method, because the FICOL density around is around 1.077 gram per ml. Okay, so the density of PBMCs are almost equal to this 1.007 gram per mole. So that is the reason these two components are coming in the same. Okay, whereas RBCs or erythrocytes, whichever the cells, those things are having more diameter or more density than the this histopac. So that's why it is sedimenting inside the tube. Then the ficol will be there. Then above that, which is very less dense PBMCs are there. Then finally plasma contents, which might be having the thrombocytes or cytokines, hormones or electrolytes will be there. So this is around very less than 1.077 gram per ml. Okay, so that is the reason people would be using this technology to isolate the PBMCs, that is called peripheral blood mononuclear cells.
Mononuclear.
Okay, so why do we need this technique to separate? Because most of the immunology related research would be done on these kind of cells, lymphocytes or monocytes or dendritic cells. So that is the reason professor have given this thing, and this is a well known technique in any immunology based lab. In our lab also people are working on this PBMC isolation, and in any immunology related study, finally the study would end with this PBMC validation. Okay, if I'm finding out any drug, then I have to isolate the PBMC from the patients. Then I have to check the what is the property of the drug if I'm giving any virus infection. Those kind of things will be in the later part of my PhD or something. Okay. And in a clinical setting, this is not exactly clinical setting, I say in a hospital setting, people will use this leukapheresis to isolate this peripheral blood mononuclear cells. This is quite tricky, and then the vein type should be in this kind of shape. Only then you can isolate this PBMC from the donor. The blood donation can be given by any donor, but that this leukapheresis donor should have this kind of setting. This Tirisual kind of vein should be there. That is what when I was trying to donate this thing they told, because I don't have that kind of structure in my vein. So they said I'm not eligible as a donor. So if you have this kind of vein structure, then you are eligible for this leukapheresis as far as I remember. So finally, he talked about the granulocytes. Granulocytes are category of white blood cells, and then which has a granular cytoplasm. That's why it's called as a granulocytes. And the basis based on their property, which when we give any dye, that kind of uptake of the dye property, it can be divided into three types. One is a neutrophil, which can be dyed with two kind of dyes, which can be anion or cationic dye. Then the basophils and then the eosinophils. So in this neutrophils as well as eosinophil, these two people are having the phagocytic property. Okay, the basophils does not have phagocytic property. Okay, phagocytic means you know now what is the meaning of phagocytosis? Phagocytosis. What's in a layman term you say? What is a phagocytic property?
Engulf.
Engulfing. Okay.
Engulfing.
Engulfing. So the neutrophils and eosinophils can engulf any pathogens. So that property they have, but basophils does not have that property. Okay. So these are some important one marks, because in gate exams now, or if you write any other exam, they would be asking nowadays. They are asking multiple choice questions. So multiple current answers would be there. It's not working. Not able to admit. Yeah. My PPTs are visible actually. I don't know what is happening. Am I audible? Someone please respond.
Yes, sir.
Yeah, some error is happening here. 2 minutes wait. I'm just checking what is happening. there was some issue. That's why. Yeah, now it is working. Yeah. So then he talked about. Now my PPT is visible, right?
Yes, sir.
Yeah. So he talked about neutrophils, basically, or polymorphonuclear cells. The polymorphonuclear means it has multiple structures of this nuclear inside the cell. So the neutrophil, the name they got it. The reason is it can be stained with both acidic, which is known as a eosine, as well as a basic dyes. That is a methylene blue. That is positively charged. And if you see any kind of pus that we are getting now, that the first cell which will come into immune action is this neutrophil cells, and these are the cells which you can see it in the pus. The pus will be having the green color now. That is a reaction of this neutrophils. Neutrophils per se not have any kind of coloring property, but it will generate a certain molecules that will give this green color. Okay, that we will see it in the upcoming weeks, what kind of molecules that is giving and everything. So these granules are also, which because we have talked about the granular structure inside the cytoplasm of these cells, so that has certain molecules such as antibiotic property molecules. So we term it as defensin, spirosidins, and then cathalsidins, bactericidin, permeability inducing protein, hydrolysis, and lysozymes will be there inside the neutrophils to kill the pathogen. And these are the cells which are around 50 to 70 percentage of WBCs in the body. And this is multi-lobed nucleus it has. That's why it is called as a polymorphonuclear cells. And these are the professional, highly phagocytic in nature. Okay. So if someone is giving any 1 mark, if what is the phagocytic nature of cells, neutrophils will be the highest. Then basophils will not come. Sorry, eosinophils, basophils will not have any kind of property. Okay, so the time limit, if you see, these are very short lifespan it has, around 2 to 3 days it will be having, and it expresses the PRRs, that is pathogen recognition receptors, that we will be seeing it in the next class, and it produces the reactive oxygen species, so that called as a free radical of the system. So this will go and kill the cell, so it forms the hypochlorite, then the hypochlorite will kill the pathogen cells. Okay, so it has two properties majorly, that is leukocytosis and then the diaperesis. And then leukocytosis is nothing but the number will increase, neutrophil number will increase upon infection. So that's why whenever you are having any kind of sneeze or any kind of infection or cough or something, then if you take your blood prick, then if you check the, if you stain it and see, then you will be seeing more neutrophils in the cells. Okay, if you are from zoology background, then you would have done this experiment in your college. So if any infection person, if you check their neutrophil count also, that will be more high. Diapedesis is such a unique terminology for immune cells, so which can cross the this blood vessels without damaging them to enter the tissue cells. So that we would be seeing it in the upcoming slides as well. And I have say already stated, if such kind of ROS production, if it is not there in the neutrophils, so what will happen, it leads to the lymphadentis. So lymphadentis is nothing but it's a swelling of lymph nodes that we have seen in the last week. Lymph nodes will be there in the all over the body. So if this swelling, if it happens, then it leads to the chronic granulomatous disease, which is called as CGD. Okay, people are joining very late. Okay, and it may also cause this CGD disease. So if you see the symptoms of this too, what will happen, the lymph node will be enlarged due to any infection or inflammation of this lymph node. This leads to the main cause of this disease, or whatever the CGD that we are talking about, the enzyme NADPH oxidase is not active here. Okay, That is the reason this kind of disease will takes place. So that is called chronic granulomatous disease. So the CGD are pediatric patients basically, and are highly susceptible to the frequent and then sometimes life-threatening bacterial and fungal infections. This infection is recurrent. So the genetic defect, if you see, that happens in the NADPH oxidase. So gene are located on autosomal chromosome. So if you see the AA disease, these people are healthy and the carrier is AA and AA disease, which means this is an example of recessive autosomal disorder. Okay, so recessive autosomal disorder means only recessive form only can cause the disease, not the dominant form of the allele. Okay, so how we will know whether whatever the neutrophils that we have it in our body is functioning or not, so or NADPH is in the function or not? There is a simple test. They use this nitro blue tetrasolium that converts this product into the blue color product. So that suggests that NADPH oxidase are normal. If not, then indicative defect in NADPH oxidase, which is called CGD disease that we say. So this is the NBT colorless product. Then if the NADPH oxidase is there, then the colored compound will be there. This we can usually see it in the normal patients. Okay. There is a new terminology. People have seen it in the netosis. Netosis is nothing but the neutrophil extracellular trap. So this induces by ROS or microbe size, microbial virulence factor, and cytokines. This composed of the chromatin, neutrophil, granular proteins, and provide high local concentration of antimicrobial proteins. Nothing but in any kind of attack or some kind of issues happen in any kind of state or any kind of village, there will be more police, right, to stop the issue. So likewise, these are the in vitro culture results. So what people have seen, when any kind of pathogen is affected in the neutrophil vicinity, there will be multiple neutrophil will be there. So that will be entrap the pathogen. So the pathogen will be cleared. So that is why it is called netosis. Nothing but neutrophil extracellular trap. Okay. So next eosinophil he took. So what is eosinophil? Which are if you compared with other neutrophil, very less in the circulation. That is 2 to 5%, and then which have the bilobed nucleus and which is stained by the eosin. Nothing but an acidic dye. That's why it is called as eosinophil. And these are phagocytic in nature, but this is not the primary function, and it's played during role during the allergic condition as well as the asthma, and it plays an important role in the parasite infection as well as increase in number and condition is known as the eosinophilia. Okay, and eosinophils, if you see, this cytoplasm also have the crystallized in nature, and it's contain the major basic protein, nothing but the MBP protein. This major basic protein, it majorly consists of toxins of element. Then it induces the histamine from the mast cells. Then that leads to the allergic reactions. So when allergic reactions are there, then eosinophils and then the mast cells number will increase. Degranulation of the mast cells will take place. That leads to the severe allergic reactions of the body. So the some protein can activate the neutrophils as well as the platelets, and it can cause the bronchospasm, which is nothing but sudden constriction of the smooth muscles in the walls. So that leads to the sneezing. Okay, whenever we sneeze, this kind of bronchospasm takes place because some allergy molecule entered our nose, so eosinophils get activated, then that causes the bronchospasm. And there are certain proteins will be there other than this MBP, that is eosinophil cationic protein, or else eosinophil derived neurotoxin also will be there to act against the pathogen. So next, finally, the basophils. Basophils are non-phagocytic in nature, and then which is also granulocytes, has the granulocyte structure in their cells, and it's least common in the granulocytes. When activated, release the histamine and other inflammatory chemicals, and it's very rare in circulation. Contains a large granules filled with the basophilic protein. So what it does usually, it binding upon binding with the IgE. IgE is a immunoglobulin E, which deals with this allergic reaction. Whenever the IgE interaction with Fc epsilon R1 receptor takes place, that releases the histamine. So this histamine increase the blood vessel permeability, then histamine is required for the smooth muscle activity. So we always know that whenever we go with any kind of sneezing issues or something, any doctor will provide the citrusin medicine. So the citrusin medicine usually blocks the H1 receptor that helps for this histamine release. So that prevent the acute respiratory distress symptoms of the body. So finally, the mast cells. So mast cells are present widely in the tissues, not in the blood thing. That's why we didn't categorize the mast cells so far. So this is a wide variety of the tissues, such as the mucosal epithelium. It will be there in tissues of digestive tract. It is there, respiratory tract, urogenital tract. It is there, skin, and then the connective tissues also. It has the mast cells. So this produces the histamine, prostaglandin, leukotrienes, which increase the permeability of the blood vessels, and it plays a really important role in case of the allergies. So any allergy, if there are people are having the food allergies or the pollen allergy or the dust allergy, so those things are comes because of the mast cells. So how it happens in case of basophils as well as the mast cells, whenever the allergen is there, the allergen specific IgE will be upregulated. Then it makes a bond with the allergen. Then this allergen IgE will make the bond with the Fc receptor for the IgE of the mast cells. Then the degranulation which takes place. When the degranulation takes place, then the hypersensitivity reaction happens. So this is called as a type 1 hypersensitivity in the body. So this leads to the IgE mediated hypersensitivity. What happened when AG induces, which means AG means antigen induces the cross linking of IgE bound to mast cells and basophils with release of vasoactive mediators. So typical manifestations include systemic anaphylaxis and localized anaphylaxis such as the hay fever, asthma, hives, and food allergies and eczema will be there. How these cells are getting activated? Whenever this allergen binds to this IgE molecule, then the IgFc receptor makes a bond with IgE. So once this happens, then the antibody cross-links takes place, then the mast cells will be activated. So this is the series of reaction takes place in the mast cells activation. Finally, we would be getting the degranulation of the mast cells. So, what is the outcome of this activation? This degranulated mast cells will secrete a number of molecules that we would be seeing it in the next slide. So, it attacks the smooth muscle cell, small blood vessel, mucosal gland, blood platelets, sensory nerve endings, and eosinophils and everything. So, this leads to the sometimes when we eat spice food also we'll get that tear kind of things now will cry, those kind of things. That's the reason because of this mast cells activation, so that leads to the sensitivity of the patient. So that's why if more mast cells activity is there, that is not good for health. That will weaken our immunity also, and then we will be very tired, and that leads to the severe reaction of the severe damage to the places wherever I mentioned here, that smooth muscle can be damaged or the small blood vessels can be damaged. That's why people are finding out drug molecule against this hypersensitivity reaction as well. So this is one such example. If you see, this is a full mast cell, and when any kind of pathogen interaction happened, then it started degranulation. Then this is fully degranulated cell. So you can see the granule structure is here now, which is not there in these cells. Okay, so there are two receptors that are really important to binding of this IgE. The one is Fc epsilon R1, another one is Fc epsilon R2, that nothing but CD23. So we will be seeing what is CD in the upcoming slides. So this is the structure of this two different receptor. One is Fc epsilon R1 and another one is Fc epsilon R2. So Fc epsilon R1 has this Ig like domains, and then the extracellular spaces, and gamma and beta domains will be there, and then the plasma membrane domain. I term will be there, then I term will create the signaling. Whereas this Fc epsilon R2 molecule, which is having the low affinity for Ig receptor and which has the extracellular CD3 soluble molecule. Then the proteolytic cleavage will happen here. Then the NH2 domain will be there. This amino terminal, this is carboxy terminal, and there are multiple double bonds also there in the structure. So, what are the secretions will be there when the mast cells are degranulated? The first set or the primary set, if you see, histamine and then the heparin will be synthesized. That is increases the vascular permeability and then the smooth muscle contraction. Serotonin will be released. That is also helps in the vascular permeability as well as the smooth muscle contraction. Then the eosinophil chemotactic factor, which means this will attract the eosinophil to the site of infection. Then the neutrophil chemotactic factor, then the neutrophils also come to the picture. Then the proteosis will be there, which means it will damage the cell wall or the cell structure. Then the secondary mediators of mast cells means there are the very low amount. What are the things will be secreted, which means platelet activating factor, leukotrienes, prostaglandins, bradykinin, and then the cytokines. Cytokines IL-1, TNF alpha, IL-2, IL-3, IL-4, IL-5, IL-6, and TGF beta and GM-CSF also will be secreted. So these are the mast cells overall. And what are the drugs people usually give to prevent the type 1 hypersensitivity? That is antihistamines, chromalin sodium. What will happen? It blocks the H1 and H2 receptors on the target cells, and nothing but we have seen the cetirizine. Cetirizine also blocker of H1 receptors on the target cells, and then the chromalin sodium, which blocks the calcium influx into the mast cells, and then the theophylline, and which prolongs high CMP levels in mast cells by inhibiting the phosphodiesterase, which cleaves cyclic AMP to 5' cyclic AMP. So this theophylline, it's always almost present in the tea or coffee as well. So coffee and tea also has the same kind of inhibition function of this phosphodiesterase. That's why whenever we drink coffee or tea, we feel really energetic. The reason is the blood glucose level will be increased due to the inhibition of this enzyme, nothing but phosphodiesterase enzyme. Then the adrenaline will be increased, which stimulates the cyclic AMP production and by binding to the beta adrenergic receptors on mast cells. And cortisone also will be synthesized, which reduces the histamine levels by blocking conversion of histidine to the histamine and stimulates the mast cell production of cyclic AMP. Then finally the dendritic cells. So dendritic cells, people when they found it out, they were thinking that it is not dendritic cell. They thought that it is a nerve cell because it has so much of dendrons, that extensions will be there. So which it is always called as a patrolling car cells, because it will patrol the everything, whether whatever it is happening. It was covered by the great scientist Ralph M. Steinman in the midst of 1970s, and he received the Nobel Prize posthumously, and he was awarded Nobel Prize in 2011. And it got originated from the hematopoietic stem cells that we have seen in the first slide itself. It can be derived from both myeloid as well as the lymphoid lineage. And there are certain set of dendritic cells present in the epidermal layer, which is known as the Langerhans cells, and which was discovered by the Paul Langerhans in 1868, but it was mistaken as a nerve cells of the epidermis. And it is also present in the gastrointestinal tract and then the respiratory tract of the tissues. So if you see dendritic cells majorly, which is having the less hydrolytic enzyme, which is compared to the macrophages, and we are going to see the macrophages in the upcoming slides, and which is having the few digestive bodies or lysosome, and then the poor viability and not so phagocytic when compared to the macrophages in vitro as well as the in vivo. In vitro in the sense in the cell culture system, or in vivo in the say the inside the body. Okay. If you want to generate any kind of this dendritic cells in the cell culture base, then you will be taking the GM calling stimulating factor. Then if you give it for another 10 to 12 days from the using the, like if you derive the bone marrows, then if you culture it using this GM-CSF, then you would be getting the dendritic cells, large number of dendritic cells you would be getting. And what this dendritic cells usually do, they take up the antigen, which induces the innate immune response, or it presents the antigen along to the MHC2 to T cells. Okay, so these are the reactions it takes place. So it participates both in the innate as well as in the adaptive immunity, majorly because innate immunity is it's a phagocytic nature, and then the adaptive immunity to stimulate the antibody response. So if you see this dendritic cells instead in the adaptive immunity, how it is happening? It has the MHC1 as well as the MHC2 receptor. The MHC1 receptor activates the CD8 cells. Then the CD8 cells will activate the cytotoxic T cells. Okay, so this will eliminate the cells from the body which are not required. Okay, then another set of cells which is called CD4 T cells, that will activate the T helper cells, which is really helpful in the case of B cell activation and antibody production. Okay, and if you see one set of plasma other set of dendritic cells, that is nothing but plasmacytoid dendritic cells, which are really important in the case of virus infection, and which induce the robust amount of type 1 interferon signal. Okay, so what will happen when the PDCs are there? It will induce the type 1 interferons are important for the antiviral state. So it will be inducing the antiviral state in terms of the innate immune response, or else it can also give the type 1 interferons as well as IL-12 and then the IL-18. Then that activates the natural killer cells. Or in the case of the adaptive immunity, it can activate through MHC1, MHC2, CD80 and CD86, CD40. Then antigen presentation will be there. There will be some promoter effector cells of CD8 plus, and then the Th1 cell response, and Treg and then Th17 response will be taken, and INKT cell crosstalk will take place. Derive of the plasma cell response. In the case, antibody will be synthesized, and immune regulation via the induction of the cell death or the suppression of T cells will take place, and finally immune cell recruitment also takes place. And there are other set of dendritic cells which are nothing but follicular dendritic cells. These are specialized antigen presenting cells which present in lymphoid follicles. And what this major component is, to bind to the to bind and retain the antigens by linking to the complement and immune complexes, then present these antigens to the germinal centered B cells. So that will be the activated B cells we say. Then finally it produce the antibody. So these are the secondary immune response that we say. So this follicular dendritic cells which rescue of bound B cells from the apoptosis and induce the differentiation of the B cells into long-term memory B cell clones, or it is called as a plasma cells. So that's why if the these memory cells are takes for so long in our body, then that helps in the antibody generation whenever the pathogen attacks once again. So this is what happens in the follicular dendritic stage. And next set of cells that we are seeing, those are macrophages. Macrophages are really good example. In the case of monocytes will become macrophage in the tissue specific cases. So the diapedesis, we have already seen it in the couple of slides back. So which means which is passage of the blood cells through unruptured wall of the blood vessels into the surrounding tissues. So this activates the macrophage by pathogens or the by cytokines also it can be activated. So whenever the activated macrophage is there, uh, that will phagocyte the pathogen, then it will be killed through the phagocytosis, the overall process. So what will happen when you compare with monocyte and the macrophage? These both are originated from the same lineage that we have seen it in the first slide. And the monocyte in this case, which are very small, less complex and less phagocytic, and these are phagos for floating phagocytic cells. But when you see the macrophages, those things are large and highly complex in structure and more phagocytic in nature and present different tissues. Keep the same system clean. That, for example, cell debris will be cleared by the macrophages, blood cells, blood clots, and then the dead cells also can be cleared by the macrophage cells. Okay, so macrophage, if you see, it has in the different status, or different body parts have different macrophages. For example, in intestine, it is known as the intestinal macrophage, or in lungs, it is known as the alveolar macrophage. If it is there, then in connective tissue, it is known as the histiocytes, and if it is present in the liver, then it is called as the Kupffer cells, and if it is in the kidney, it is called as the mesangial cells, and if it is the neuronal, it is called as microglial cells, and in bone, it is known as the osteoclast. So in vitro culture, if you want to generate the macrophages, then we would be using MCSF. It's called a macrophage colony stimulating factor. Then it would be we would be getting more macrophages. Okay. So difference between the macrophage and neutrophils, we have given it in the this slide. This would be helping you for the exam perspective. Macrophages are long-lived, which means the months to year, whereas neutrophils are short-lived, and this present in the tissues when it get mature, and then but the neutrophils are not resident of the tissue things. It is mostly in the circulation, and it always keeps in the circulating form, and it able to migrate from tissues in lymph node, allowing the antigen dissemination, whereas this does not show any kind of such properties. And the macrophages not very strong stimulant for the inflammation, but neutrophils are strong stimulant for the inflammation. And we have seen oxygen independent killing is not vigorous in the case of macrophages, but oxygen dependent killing is lethal to the microbial pathogen in the neutrophil case. And if you see, the intracellular condition may permit the growth of intracellular microbe to grow inside, such as MTB. So MTB can grow inside the macrophage itself, but it is extremely hostile for the intracellular microbe to survive inside the neutrophil. And pathogen can circumvent the respiratory bus, but pathogen cannot do this kind of things in the neutrophil. And breakdown of the phagosome takes place and prevent the phagolysis of formation and resist the release of the granule contents. But here, if you see, if the pathogen need to survive, then need to resist the phagocytosis, which cannot be done in the case of neutrophil, and kill the neutrophils or get killed. These are the only system can be taken place in terms of the neutrophils. Either that pathogen can be killed, or the cell can be killed to survive inside the host system in terms of the pathogen perspective. And what are the different reactive oxygen species people have seen? There are two reactive oxygen species. One is the reactive nitrogen species, that reactive species in this case, that is nitric oxide, nitrogen dioxide or peroxynitrate, or nitrogen oxygen species if you see superoxide anion, or hydrogen peroxide, hydroxyl radical, and then the hypochlorite anion will be there. So how these things are really synthesized in the body? When the molecular oxygen is there in the presence of NADPH and then the arginine, the nitric oxide synthase will come, then it will produce the citrulline. Then this nitric oxide will be formed here. Then the other way, how the other reactive oxygen species will form, this nitric oxide again combined with the other set of oxygen, then it would be giving nitrogen dioxide. How it happens? Whenever the two molecular oxygen combines with NADPH, NADPH oxidase will form free radical of oxygen. So then it binds to this nitric oxide, then it will be forming the nitrogen dioxide or peroxynitrate. But in this case, the superoxide dismutase that forms the hydrogen peroxide, which combines with this chloride radical to form hypochlorite. Hypochlorite is nothing but a bleach solution that we usually use to clean the restrooms and everything. But this also synthesized in our own body itself to kill pathogen. So then he was talking about the natural killer cells. So natural killer cells overall, if you see, which is around 5 to 10 percentage in the blood lymphocytes, and this is also the granular lymphocytes. And this granule contains the perforin and then the protease. Otherwise, we call it as the granzymes to kill the pathogen. Okay, and it induces the pore formation in the target cells, and then they trigger the apoptosis. And this natural killer cells have the cytotoxic properties against the tumor as well as the virally infected cells through ADCC. It's nothing but antibody dependent cell mediated cytotoxicity, we say. How this NK cells are having these properties? Because many of the cells can also become a tumor cells in a normal life itself, but this NK cells will identify those cells, then it will be removed from the system. Otherwise, most of us will become a tumor patient so far. So this antibody dependent cell mediated cytotoxicity exclude the FC gamma R3 receptor or CD16 receptor. It takes place. How it happens? Whenever the NK cell is there, it can sense the this MHC1 receptor. This MHC1 molecule will always there in all nucleated cells. Okay, so if it is not having, or if the amount is very less, then it will become the unusual profile of the MHC1. Then it can be eliminated from the system. Okay, the NK cells usually that are activated by different mechanism, that is IF and gamma, IL-2, IL-12, and then the IL-18 also can uh, uh, activate the NK cells. And how we will see that whether this NK cells are working or not, there is a assay called chromium release assay. So the radioactive labeled chromium will be used. So what will happen? Na2CrO4 will be introduced in the medium. Then if the NK cells are added, then it will convert into Na2Cr2O7. So that will be released the chromium in the supernatant. And if that mechanism is there to detect the radioactive material, then we can say NK cells are active. Okay, so the factors affecting the this ADCCs include density and then stability of the antigen on the surface of the target cells, and then antibody affinity, FCR binding affinity. So there are other subset of cells, that is the natural killer T cells, which is a subset of T lymphocytes we say, and which has the both a function of NK, that is natural killer cells, as well as the T cell property. How it happens? This shares the certain surface markers of both NK as well as the T cells. This NK T cells express the T cell receptor that interact with the glycolipid antigens. So this NK T cells are always said as non conventional antigen detection. Antigen detection. Okay, non-conventional antigens are detected by the NKT cells, including the glycolipid. So the marker is CD1D. So this is really important in CSIR exam. There are more exam they will be asking. Glycolipid is detected by which receptor or MHC related molecule, that is CD1. Okay, on the antigen presenting cells. Okay, this get activated during the infection and then during inflammatory condition as well. Upon activation, the cells produce the various immunomodulatory cytokines and they influence the other cell types. So it plays really important role in the case of infectious agents, autoimmune disease, tumor, as well as in the allergens case. So this is one such example. In this antigen presenting cells, this can be detected by, or it what we say, presenting the CD1 through CD1D things along with the MHC one molecule. It is presenting the glycolipid to the NKT cells. So when it presenting the NKT cells, then it can also give the Th1 cytokine response, that is IF1 gamma mediated, or Th2 cytokine response, which is IL4 mediated. Okay, so sorry, yeah. Next cells are B cells majorly. So these are called as a B lymphocytes otherwise. So in the prenatal stage, these are coming from the fetal liver, and later after birth, these are coming from the bone marrow or in human. Okay, and it is the central to adaptive immunity because it helps to synthesize the antibody needed for the pathogen neutralization, and responsible for the production of antigen specific various types of immunoglobulins, such as otherwise known as antibodies. The function of B cells was discovered in 1960 by Max Cooper, and he demonstrated that antibody production was completely abrogated in chickens after the surgical removal of bursa of fabricus. That is why it is called as B cells, because this bursa of fabricus was not there. So that's why it is called as B cells. So therefore, this B alphabets are derived from this bursa of fabricius. Okay, and B cells are needed for the CD4 T cells mediated response. and it is not essential majorly in the case of CD8 plus T cell response. So we have been seeing so many of CD. What is CD in the sense? Cluster of differentiations. So what is cluster of differentiation? There are specific surface protein which are present in the different cells that is unique property to those cells only. So how people have understood this thing? These are functionally different and then different surface protein, and these surface proteins are cells surface receptor signaling or the adhesion molecule. Additionally, the same cells express the different surface proteins during the development process and during the activation as well. Overall, these surface proteins are called as cell surface marker. Okay. And these cell surface markers are detected by the monoclonal antibodies. Monoclonal antibody means which would be detecting only one set of uh, antigen or the epitope on the antigen. Okay. So that's why it is specific to CD. And then the cell surface protein to which these set of antibodies are bind are called CD molecule or marker or antigen. So the CD molecules has a biological significance. In the case, it can be used for the isolation, for the characterization of the particular cell types, or even therapy. The CD marker can be used for diagnostic purpose as well. The nomenclature CD is done along with the number. It represents the order of discovery. So the CD1 is there, it discovered in the long back in the first case, and then CD4 discovered later, and CD8 discovered very later. Okay, and as far as the number concerned, around 400 different series are there in the body so far. Okay, so these are the different markers for the different thing. So if you see the hematopoietic stem cell which is having the CD45 RA minus, CD48 49 plus, and CD90, this one. Then if you see the MPP, then that is also having the CD45 RA, then the CD90 or TH. So like this, if you see multiple blood cells would be having the multiple markers for specific CDs for their identification, so which would not be there in the other set of cells. Then finally the CD for T cells, or the we are talking about the overall T cells. So this T cells would become different CD cell T cells. For example, TH22 cells, which would be mainly focused in the case of the mucosal immunity, inflammatory diseases, and the barrier defense in the case of the infection. There are T17 cells which induce the inflammation and then which promote the autoimmune diseases in the body, and Th1 cells which helps in the intracellular pathogen elimination, and there are Th2 cells which induce a strong antibody response and then the allergic response in the case, and then there are Treg cells which maintains the self tolerance against the our self cells, and then the Th9 cells which helps in the humoral immunity through the B cell interactions and functions on the many cell types and including the mast cells and other CD4 T cells and the anti-tumor. And there are TFH cells, and which are specific to the antigen specific B cell immunity. So that is what I have for the lecture. If you are having any doubt, then we can discuss in the thing, or we can go to the assignment session. So which one we can do first? Do you have any specific doubt?
No,
No,
sir.
sir.
Okay, then we can go to the question and answer session.
Yes, sir.
Yes, sir.
Yes, there it is. Yeah. So what is the approximate number of hematopoietic stem cells in bone marrow? This I have not taken, but still the question is there. So the answer is 1 in 50,000. Okay. This we have seen it in the last to last class. So it's very less hematopoietic stem cells are there in the body. So what is the markers of this hematopoietic stem cells? That is CD34 and then SCA1. This we have seen. If the bone marrow cells are cultured with MCSF, it will differentiate into what is the full form of MCSF? What is the full form of MCSF? You can tell if you know, you can tell, or else I would suggest what it is. What is MCSF? It's called macrophage.
Stimulating
That is as for
macrophages colony
yes, macrophage colony stimulating
stimulating factor.
factor. So when you take bone marrow cells, then if you culture it with MCSF, then it will get into macrophage. Correct? Okay. This PDCFLT3 is okay. Okay, we'll go to the first, this fourth question. To make PDCs, plasmodic dendritic cells from HSCs, we would be using FLT3. I didn't tell this thing in the record lecture. Okay, which of the following immune cells is formed through both lymphoid and then the myeloid lineage? What is the cell line?
Dendritic
Dendritic cells. Okay, okay, okay. If NBT test was performed, did not result in the blue color complex, it is indicative of what? That blue color, so test we have seen, na? NBT molecule, it adds with which enzyme? One enzyme we were seeing, na? That is NADPH oxidase. If it is functions, then it will give the blue color, right? But here, if any kind of chronic granulomatous disease, if it is there, then the NADPH is defective. So that is the reason the chronic granulomatous disease is the answer. Eosinophils are having the major basic protein, nothing but which is a MBP. Yeah, tell me. Which of the following statements is are correct? First option. Are incorrect they are asking. Okay, which is not correct they are asking. So the antibodies are predominantly produced by erythrocytes. Is it correct? Erythrocytes produce antibodies.
No.
No. So which one produce antibody? Which cell produce antibody? B cells or B lymphocytes. Specifically, if you say plasma cells. Okay, which means this is a wrong option. So which means the answer for this question. On maturity, the blood cells do not lack a nucleus. Is it correct statement? Mature red blood cells, it lack a nucleus or it does not lack a nucleus. Which one is correct? On maturity, the red blood cells do not lack nucleus. Is it correct? Is it a correct statement? Yes or no, you tell, then I will answer it. Na?
No.
No. Because it does lack a nucleus when it is on maturity. So the white blood cells have a lesser lifespan compared to red blood cells. Is it correct?
Correct, sir.
Correct. So what is the lifespan of red blood cells? 120 days. Okay. Whereas white blood cells would be having less than 1 week. So which means which are incorrect they have asked. So which means 1 and 2 are incorrect. Okay, next question. Which of the following statement is are correct? They asked. Okay, macrophage mature into monocyte and migrate from the blood to tissue. Is it correct? Will macrophage mature into monocyte? You can tell any answer. I will correct you. Don't worry. If it is wrong or right, don't worry. Is it correct? Macrophage mature into monocytes. Anyone is there? As if I am only talking. Other people are there. Vaishnavi, Revati.
Correct.
Is it correct? How? See, monocyte will become macrophage. Okay, because monocyte are
Sir, wrong,
in
sir.
blood, and these things are in tissue. So that's the reason. This is correct. Migrate from blood to tissue. It is correct. Okay, but monocyte is mature into macrophage, not macrophage into monocyte. So which means it is wrong. So that is not the correct answer. B lymphocytes comes under the granulocytes category. Is it correct? Granulocytes are only neutrophil, basophils, and eosinophil. So B lymphocytes are granulocytes are what? No. Okay. Megakaryocytes are produced from the blood platelets and are released into bloodstream. Is it correct? Is it correct? Blood platelets are
No, sir.
produced from megakaryocytes. Okay. So which means all are none of the statements are correct. All are wrong. Okay. Okay. Last question, then we can wind it up after this. Neutrophils and basophils are predominantly involved in the phagocytosis of pathogens. Is it correct? Neutrophils and basophils. We'll go back to the thing. Wait. Then you will understand. What it has written here. Neutrophils and eosinophils, right, are phagocytic pathogens, which means, is it a correct option? Neutrophils and basophils. This is should be eosinophil. So which means this is a wrong statement. Leukocytosis is associated with a decrease in neutrophils upon infection. Is it correct?
No, sir.
Which means what is the answer then? It is increase. Okay, increase in the neutrophils upon infection. So that is also not correct. Chronic granulomatous disease is a genetic disorder. Is it correct?
Correct, sir.
Okay, then which option is correct?
Option C.
Yes, option C is correct. So C only. Yeah, that is what I have for you people today. If you want to ask any question other than this, then you can ask. Like other than this, like if you want to ask about subject, any other subject, or exam or something, then you can ask. Any questions you have? Okay, I think there are so many new people are there. Can you please introduce yourself? Aswini, Ema, Revati, Pranita. I just want to know where are you from? What are you people doing? Can you please introduce yourself if possible? Or you can use the chat box to type as well. Hello Ashwini, where are you from? And what are you doing? You're studying which course? MSc Microbiology. Which year? First year or second year? Good, good, good. So Paldikonda, it's from Andhra or Telangana? Okay, Pranita studying MSc Biotechnology, Andhra University, Visakhapatnam. Good, good, good. Vellore district. Okay, so you're from Tamil Nadu, Ashwini. Good, good, good. So what's your future plan after your MSc? I will share it on the. There is a link na in the this thing only. Your NPTEL classes. There it will be uploaded. I'll show where how to do it. This online courses na. I'll show. You know na there is a one session called doubt clearing session. It will be there. I'll show. So what do you want to do, Ashwini, Subulakshmi, Pranita, after your MSc? What's your plan? I think you should go for internships now during your summer vacation now. There are most of the internships are open, so you people are eligible to apply. Wait, I'll show where to. Yeah, this is the course thing, right? Here, if you see problem solving session, Jan 2025. So if you click this, then all the recordings will be there, including the videos, all those things. We will upload it here. Okay, so you can go back and then check it. Okay, Subbalakshmi.
Okay, sir. Thank you, sir.
So you appearing for any internships or what? Because now some IIT announced internship for students. So if you people are interested, then you can go for it. So you people are coming under which university actually? This Vellore, you said na? Which university? You said you
Tiruvallur University, sir.
Tiruvallur University. It's a state university, right?
Yes, sir.
Good, good. So what are the subjects that you people have now? Like like core subjects I'm asking this semester.
Medical bacteriology and mycology,
Okay.
medical virology and parasitology,
Okay. So you are from microbiology background or biotechnology? Which background you are from?
microbiology, sir.
Microbiology. Okay. Okay. That's why so much of practicals wise. What are the topics they teach you in this semester?
Practical.
Have they started or still it is going on? Have you gone through any practical classes as of now?
Yes, sir.
Okay.
MIC,
Okay.
minimal incubatory concentration,
Okay. What experiment you did?
minimal bacteria.
What is MIC? Minimal inhibitory concentration. Na? What is the meaning of it? Good. Okay. Whenever you study, you people study it clearly because that will help you to get into any interview. Because in your interviews also they would be asking these questions only: what you have done, what are the practical classes that you have gone through. I mean, if you want to become any kind of a faculty or if you want to get into any kind of lab also, they would be asking it from your practicals only. Understood?
Okay, sir.
Yeah. So Pranita, uh, you are from which background you said? MSc Biotechnology. Okay. So in University of Hyderabad also there are multiple faculties are from your university. They studied MSc in your university. They did a PhD in University of Hyderabad or somewhere. Then they got postdoc abroad. Then now they are great faculties here. I most of the faculties are from here only. Good, good. So overall class, how was it? If you have any suggestions, if I want to improve something, then you can suggest me that. Then I will try to incorporate those things. Overall, how was it? Any critical suggestions if you want? If you are not able to understand something, then if you tell me, then I can improve it in that way.
Nothing, sir.
Nothing. Nothing. It's not nice. How was it? You can give your feedback at least, na? How was the classes? Was it good or was it understandable?
Good and understandable, sir.
Good.
Explanation is very clear, sir.
Explanation?
Explanation is very clear.
Okay, good, good. So what I would suggest, yeah, weekly once we would be having the same class. So next week also I will start around 7:15. Then we will have 1 to 1.5 hours of like 1 hour of class. Then we would be discussing around half an hour. And not only this subject, if you have any, yeah, thank you so much, Parnita, right? Pranita. So what I would suggest, if you have any other subject doubt, you can ask me because I have been teaching for different subjects in NPTEL itself. So if you have any other subject doubt also, then I can help you. If you want to write talk about CSAR or JRF exams, GATE exams, so I can guide you that how can you clear.
Okay, sir.
Yeah. So Kritika, where are you from? I think you didn't introduce yourself if I'm not wrong. Yes. Kritika, are you there?
They are also coming from Vellore,
Oh,
sir.
you are. So most of you are your class only.
Yes,
So
sir.
Hema is also from your class. Ashwin is also from your class. Good, good, good. So this is compulsory or how it is? Like, will they give any score or grade for this thing? Or what is the criteria in your college? Because there are certain colleges they would be giving marks for this or some grade credit points they give.
Credits.
Hmm?
Credits, sir.
Okay, how many credits that they give?
2 credits,
Oh,
sir.
this is 2 credits. Okay. It's compulsory for all MSc students.
Yes.
Good, good, good. Other than this, you would be going for on-job training or somewhere you should go on a 15 days training. Project would be there because during my masters, my UG college, they have these components, projects and everything. You people do have those kind of things. Do you have those kind of things in your curriculum? Good, good.
Internship compulsory, sir.
Internship is compulsory. So where are you going to do, Ashwini?
Not yet planned, sir.
Good. So where did you people do BSc then? Because I'm also from Tamil Nadu
Same.
actually. I did my bachelor's in Coimbatore. PSG College of Arts and Science, you people know? Bardiyar University, PSG College of Arts and Science.
VIT, sir.
Okay. No, no, I did my bachelor's from Coimbatore.
Okay, sir.
Okay. So then I
Can
joined.
you apply for fellowship, sir?
What ma?
Fellowships, can you apply, sir?
So you're asking, do I have any fellowship?
Yes, sir.
Okay, I have a Prime Minister Research Fellowship, which is the highest paid
Okay,
fellowship in
sir.
India. So
Okay, sir.
I'm getting around 75,000 per month as a salary,
Mm.
and 2 lakhs per annum for my research. And I have cracked CSIR twice, DPT twice. I have cracked day gate. I have cleared TIFR exam. I have cleared National Institute of Immunology exam. Those many things. And here also I am doing my integrated PhD course. This is also entrance based. So I have cracked entrance. Then I have gone through this interview process. Then after my BSc, this happened. So joining here as an integrated student. Then I joined.
Sir, any internships are available?
In our university, you're asking.
Anything, sir.
Okay, so I would suggest that already when I started the lecture, there is one website called Biotechnica. There is a website,
Yes,
so
sir.
their internships options will come. You can write, or you can write it to the faculty directly by writing to them that don't write that first read their papers, one or two papers. Then if you get the interest, then you highlight those things. I read these two or three papers. I want to learn about these techniques from your lab. That would be helping for me to do my like to continue in this field as a researcher. Those kind of things you have to write. And for you people, I think in Chennai, there is one institute, ICMR Institute, the Tuberculosis Institute is there, right?
Yes,
There
sir.
it's free of cost actually to do internship.
Okay, sir. Any
I
applications
think you
are
have to
there,
write
sir?
it to the director directly through your HOD, if I'm not wrong. You have to write a request letter through your HODs. They have to sign it. Then
Okay,
you will
sir.
send it to them. They will give you for everyone they are giving. So you can go to that institute. And Adayar Cancer Institute is there, na? There also they are taking
Are you?
Adayar Cancer Institute, Chennai.
Okay,
So there
sir.
also they are taking students. No, sir, I want to do it in a very good place. Means then you have to approach the faculty directly. IIT Madras, if you wanted to do, then you have to write it to the faculty directly. Then they would be taking you for the internship. Yeah. So you
Okay.
have to write the para in such a way that SOP should be there. That statement of purpose, why you want to do internship there, how will it be helpful for you, or in the current scenario or in the future. They would be telling those kind of things. So that would be helping you for in a larger perspective, I would say. So better you can read.
In Chennai,
Hmm.
Cancer Institute. Another one, sir.
ICMR Tuberculosis Institute. ICMR Tuberculosis
I,
Institute.
okay, sir.
There are multiple institutes are there. You can go anywhere and then finish it. I think 15 days you have to go, right? 15 days or 1 month. How many days they are telling in your college?
20 days, sir.
20 days. You can easily apply. Then you can go and come back. Any other
Or 1 month or all.
it is up to you, but I would say that you can go. There are certain things. One more thing I forgot to tell. There is a one visiting scholar thing is there. I will tell you right now. Please stay. One more thing called TIFR, Visiting Scholar Program, VSP program. TIFR. I'm typing it. You can
Okay,
take a
sir.
screenshot. TIFR, VSP, VSRP. Now they are giving you 15,000. These are they will give you the money. Okay, I'll show you.
Okay, sir.
I've checked it. Recently, they have announced actually. You would be getting around 10,000 or 15,000 per month to do your work there, and they are giving free accommodation and everything. And you need a good recommendation from two faculties or one faculty should be there. Then, if you give nice SOP, why do you want to do uh, internship there? They would be giving. This is the one. See, eligibility is student of any stream in their final year of BSc or final year of their MSc, pre-final year. Pre-final year means first year. Okay. An integrated MSc, four
Yes,
years
sir.
of MSc. So you people are eligible to apply.
Yes, sir.
So application process, please apply directly by contacting the respective faculty member. So you can apply now itself.
Okay, sir.
It's really good that you
Okay,
would be
sir.
getting the see stipend how much they are giving. Selected students are paid a monthly amount of 9,000 and travel reimbursement. So if you are traveling by train or something, that also they will give. Then the free accommodation also they are giving. This much they are providing now. So last date is
Okay, sir.
28th of this month.
Okay,
So
sir.
you can take a screenshot of this website as well. So that would be really really helpful. Multiple things will come like this.
O-
If you go to this Biotechnica website now, you have to check it continuously. What is coming? How can you apply? Because these are very competitive now. Multiple people will be applying from different areas. See here now internships biotechnica.org.
Yes,
So
sir.
there you will be seeing what is the recent thing. See short-term training internships at NCCS on SIR project, summer research internship in IIT Gandhinagar. For IIT Kanpur also they are asking. So these kind of things will be there. You can apply for it, then you can go and do it. Or else, if you want to go and stay there for some days, then you can write it to them directly. Then also you can go. That also will be helpful for you.
Okay.
Understood. Any other doubt you people
Okay,
have?
sir.
Ashwini, do you have any other doubt?
No, sir.
No doubt. Okay. Good. All the best. Do well. Next week we will see the same time. Okay.
Thank you,
Now
sir.
you people can leave.
Okay, sir.
Good night.
Good night, sir.
Thank you. No mention Hema. Good night. Take care. You people can leave. I will leave around 9 o'clock. Another 4 minutes. I'll wait, then I'll leave.
Okay, sir.
How will we contact you? I will give you my mail ID. Wait. Yeah. This is my mail ID, siranjeevirangaraj@gmail.com. So you can copy it and then keep it with you. If you want to contact me, you can contact through that. Bye, Subhlakshmi. Take care.
Bye, sir. Thank you,
Yeah,
sir.
bye.
Okay, sir.